red button at the top. This one. Here it is. There's overflow and the big blow. That's right down there. It might be full too. Yes. Hi.
Live from the North Carolina Museum of Natural Sciences in beautiful downtown Raleigh, it's the Science Cafe with your host, Chris Smith.
Well, good evening, everybody. Let's get started. Welcome, everyone, to the Science Cafe. My name is Chris. I am your host for our Thursday night programs here at the Museum of Natural Sciences. Welcome to the museum. I am so glad to see so many people interested in the brain. Raise your hand if you've got one. Okay. Just saying, I got family. Yeah, welcome to Brain Awareness Night here at the museum. This is an annual event that we are fortunate enough to host here at the museum, where we bring in neuroscientists from the area and from abroad to make us aware of our brain. Raise your hand if you had the opportunity to actually hold one of the brains in tonight's program. Yeah, so for tonight, right after the cafe, as soon as we're done here, you'll be able to go into the first floor of the museum and actually see some of the exhibits and meet the exhibitors who have come out, local scientists and local labs doing lots of interesting work, and you might get the opportunity to actually hold an actual brain. I had the chance. It was really cool. You can see pictures on the museum's Instagram account. If you don't get the chance, you can at least see it on Instagram. And the only thing I could think was that all of our thoughts, ideas, all the great things we do, all the terrible things we do, all the sort of middle in-between things we do, I was holding in my hand. The thing that thinks those thoughts, and then the thing that tells my body to do or don't do those things, right, was just a couple, 3 pounds of stuff in my hands. It blew my mind because, well, isn't that funny? But look at that. Our vernacular carries with it these incredible things associated with our brains. So tonight's program is a collaboration. I do want to mention, and I want you to applaud for the Keck Center for Behavioral Biology for bringing in tonight's speaker and working with the museum on tonight's event. Give it up for the Keck Center. Absolutely. Thank you very much. Tonight's guest is Dr. Jeff Lichtman. He's from Harvard University. He has an incredibly distinguished career of neuroscience work. You've probably seen something that his lab has worked on, even if you didn't know the name. So without further ado, put your hands together and welcome to the Science Cafe, Dr. Jeff Lichtman.
Thank you. Thank you very much. It's a great pleasure to be here and to talk to you about my favorite subject. And I think it's fair to say that probably everybody here, even though many of you are not biologists, know what vital organs are. And maybe can we have the music? Everybody knows what vital organs are, and we have a bunch of them. You have your liver, your kidneys, your lung, your heart, and of course your brain. And I would say, without exception, all of you really like your brain. And although you have these other organs and you're using them, if you went to your doctor and your doctor said, your kidneys are shot, I'm going to have to get you a kidney transplant and get you a new kidney, you'd probably say, okay. And if somebody said, your heart is shot and it's really important, I'm going to have to give you a heart transplant, you'd probably say, yeah, okay. If I said, your body's great, but your brain, it's shot. But I can put a whole new brain into your body. Chances are you would say, nah, I'd prefer to keep my brain. And there is an interesting reason why you have more sort of chauvinism for your brain than you do for your kidneys. And that is because when you think of yourself, you think that everything you think about yourself, all those things that make you a person, is not related to how well kidneys work, although they are vital organs. If you don't have kidneys, you will die. But what your brain does is it records all those things that happen to you. It is basically you. So if I had to take your brain and put it into a new body, because your body was shot, you'd probably take that, but you wouldn't take a new body, a new brain on a perfectly good body. You want your own brain. And I think a deep mystery for neuroscience is how all this information about the world gets stored in our brain. What actually happens during experience that turns you from a baby that knows nothing about the world into the magnificent creatures you are as you grow up and learn a language and learn to use tools. And rather than sort of pontificating on this point, I'd like to start by showing you a video from YouTube. Some of you may have seen this before. It's quite a nice video, and I wonder if the people working the magic in the back can start the video, the YouTube video right at the beginning with sound.
Hey, it's me, Destin. Welcome back to Smarter Every Day. You've heard people say it's just like riding a bike, meaning it's really easy and you can't forget how to do it, right? But I did something. I did something that damaged my mind. It happened on the streets of Amsterdam, and I got really scared, honestly. I can't ride a bike like you can anymore. Before I show you the video of what happened, I need to tell you the backstory. Like many 6-year-olds with a MacGyver mullet, I learned how to ride a bike when I was really young. I had learned a life skill, and I was really proud of it. Everything changed, though, when my friend Barney called me 25 years later. Where I work, the welders are geniuses, and they like to play jokes on the engineers. He had a challenge for me. He had built a special bicycle, and he wanted me to try to ride it. He had only changed one thing. When you turn the handlebar to the left, the wheel goes to the right. When you turn it to the right, the wheel goes to the left. I thought this would be easy, so I hopped on the bike ready to demonstrate how quickly I could conquer this.
And here he is, ladies and gentlemen, Mr. Destin Salem. First attempt riding the bicycle.
All right. So the faster I go, the better. Yeah.
I couldn't do it. You can see that I'm laughing, but I'm actually really frustrated. In this moment, I had a really deep revelation. My thinking was in a rut. This bike revealed a very deep truth to me. I had the knowledge of how to operate the bike, but I did not have the understanding. Therefore, knowledge is not understanding. Look, I know what you're probably thinking. Destin's probably just an uncoordinated engineer and can't do it. But that's not the case at all. The algorithm that's associated with riding a bike in your brain is just that complicated. Think about it. Downwards force on the pedals, leaning your whole body, pulling and pushing the handlebars, gyroscopic precession in the wheels. Every single force is part of this algorithm. And if you change any one part, it affects the entire control system. I do not make definitive statements that often, but I'm telling you right now, you cannot ride this bicycle. You might think you can, but you can't. I know this because I'm often asked to speak at universities and conferences, and I take the bike with me. It's always the same. People think they're going to try some trick or they're just going to power through it. It doesn't work. Your brain cannot handle this. For instance, this guy. I offered him $200 just to ride this bike 10 feet across the stage. Everybody thought he could do it.
No, you didn't understand. You didn't understand. So, this way. All right, so, whenever you're ready. Yeah. Quickly! No, you have to keep your feet on. Dude, all right, here we go. Wait. Like, you gotta start rolling at least. And go. Oh, no! All right, back up. Okay, wait. Keep your feet on the pedal. Go. Go right off. Just keep your feet on the pedals. Yeah, come on.
Once you have a rigid way of thinking in your head, sometimes you cannot change that, even if you want to. So here's what I did. It was a personal challenge. I stayed out here in this driveway and I practiced about 5 minutes every day. My neighbors made fun of me. I had many wrecks, but after 8 months, this happened. One day I couldn't ride the bike and the next day I could. It was like I could feel some kind of pathway in my brain that was now unlocked. It was really weird though. It's like there's this trail in my brain, but if I wasn't paying close enough attention to it, my brain would easily lose that neural path and jump back onto the old road it was more familiar with. Any small distractions at all, like a cell phone ringing in my pocket, would instantly throw my brain back to the old control algorithm and I would wreck. But at least I could ride it. My son is the closest person to me genetically, and he's been riding a normal bike for 3 years. That's over half his life. I wanted to know how long it would take him to learn how to ride a backwards bike, so I told him if he learned how to ride a backwards bike, he could go with me to Australia and meet a real astronaut.
Are you going to give up? No. Go ahead. This is how it starts. Look at this. This is such a big deal. Get up, you got it. Did you see his brain get it? So he, in, how many weeks have we been doing this? 2 weeks? In 2 weeks, he did something that took me 8 months to do, which demonstrates that a child has more neuroplasticity, am I even saying that right, than an adult.
It's clear from this experiment that children have a much more plastic brain than adults. That's why the best time to learn a language is when you're a young child.
All right, today's bike log. I can ride smooth, I can ride fast. I'm thinking the experiment is over.
Okay, now I'm in Amsterdam, a city that has more bicycles than people. The question is, can I ride a normal bike now? I mean, I've spent all this time unlearning how to ride a bike. If I go back and try to ride a normal one, will my brain mess up? So I've tweeted a Smarter Everyday meetup, if you will, and I'm going to see if somebody brings a bicycle, and I'm going to try to ride a normal bike. It's backwards.
This was one of the most frustrating moments of my life. I had ridden a normal bike since I was 6, but in this moment, I couldn't do it anymore. I had set out to prove that I could free my brain from a cognitive bias. But at this point, I'm pretty sure that all I proved is that I can only redesignate that bias.
So what you're not seeing is just a group of people here looking at me, looking at the strange American that can't ride a bike because they think I'm dumb. But I'm actually 2 levels deep into this because I've learned and unlearned. All right.
After 20 minutes of making a fool out of myself, suddenly my brain clicked back into the old algorithm. I can't explain it, but it happened in a very specific moment.
I got it. I'm back. Oh, it clicked. Hold it, it clicked. I got it. Okay, there it is. There's the moment. Okay, I can ride a bike.
I tried to explain this to the people around me, and they just didn't get it. They thought I was faking the previous 20 minutes, and I couldn't get anybody to believe me.
That looked like I faked that, didn't it?
Yeah.
Just a fake. You think I'm faking. You don't believe me.
This is so weird. You're like,
You think I'm lying, don't you? I'm not lying.
I felt like the only person on the planet who had ever unlearned how to ride a bike, and I couldn't articulate it to anyone because everybody just knew that you can't forget how to ride a bike. So I learned 3 things from this experiment. I learned that welders are often smarter than engineers. I learned that knowledge does not equal understanding. And I learned that truth is truth, no matter what I think about it. So be very careful how you interpret things, because you're looking at the world with a bias, whether you think you are or not. I'm Destin. You're getting smarter every day. Have a good one.
Okay, if you want to support Smarter Every...
Okay, so thank you for listening to this interesting example. And now I want to talk about the neurobiology behind what's going on. And honestly, we don't know completely, but I want to tell you a little bit about the way the nervous system is wired up and the way that wiring could change. And I'm going to talk about a particular muscle that I've studied for about 30 years, so I know more about this muscle in the neck of a mouse. This is an anesthetized mouse whose neck has been opened up and through a midline incision. The animal is sleeping, intubated. That tube coming out of its throat means I'm breathing for it. And I've made a midline incision in the neck, and I've pulled on one side a gland that secretes saliva, the salivary gland, to the side to reveal this big muscle. You can see that big thing with the yellow arrow pointing to it. That's the sternomastoid muscle. This is a muscle humans have, and if I had a better neck, that is a bigger neck and less fat, you could see it on me. And if any of you are thinner than me, which is virtually everybody here, you can feel this muscle that goes from the mastoid process beneath your ear all the way down to your sternum. And ballerinas and athletes, often these are really impressively bulging muscles. It's a very nice muscle because you can see without doing any further dissection in a living animal where the nerves from the brain that activate this muscle are. And they're in that little area right in the middle of the muscle with the sort of zigzags. I don't have a laser pointer that I can use for this, but we've made a mouse by genetic engineering where all the nerves glow yellow. And I'm going to show you what that muscle looks like in this fluorescent transgenic mouse in this following slide. So that little area with the little red box is there. So when you light up the nerves, you see this extraordinarily nice-looking tree-like structure. And it looks like there are little red leaves at the end of the branches. Those are sites on muscle fibers that respond to the chemical the nerve is releasing to make muscle fibers twitch. It's called acetylcholine is the chemical, and those red things are acetylcholine receptors. But that's beside the point at the moment. At the moment, I just want you to see this massive nerve whose job is to make the sternomastoid muscle contract when you want to turn your head one way or the other. To get a better sense of actually what nerves look like, I'm going to zoom in on a little part of this tree, that little area that's boxed in there, and show you that with higher resolution microscopy. And now you see that that yellow branch is actually made up of a bunch of individual yellow fibers. And those fibers are coming from nerve cells in the spinal cord that are making connections on muscle fibers at those red things, which are called neuromuscular junctions. They're the junction between the nervous system and muscle fibers, and they are synapses. They're where nerves talk to muscle by releasing a chemical called acetylcholine that binds to an acetylcholine receptor, which is labeled in red. And that's why there's red under the endpoints of each of those branches. And one thing I want you to notice here is that each of those red pretzel-shaped objects has only one nerve fiber connecting to it. These nerve fibers are called axons. I'm going to test you later on this, but so you may want to remember that. But these axons make one connection per neuromuscular junction in adult muscle. But if I wanted to ask which particular fiber, which particular one of these axons went to each of those neuromuscular junctions, it's a little tricky because all of the axons are exactly the same color. And if you count the number of axons in that bundle coming up there, there are fewer axons than the number of muscle fibers, so they must be branching. There must be branches of those axons going to more than one. But to see all of them, we tried to come up with a technique that would put a different color, a different fluorescent color in each axon. These fluorescent colors, this yellow color, comes from a jellyfish, and it is called yellow fluorescent protein, and it's a gene in a jellyfish that we engineered into a mouse. So this is a very weird kind of biology where you're putting jellyfish genes into mice to make their nerve cells glow. That's what we did. But then we realized that there wasn't just this green or yellow color that we could get out of jellyfish shown in the middle, but there are all these other fluorescent proteins from the ocean. Some of them are red, and they come from corals. Others are blue, and they also come from corals. So we had red, green, and blue colors. And does anybody know why that's significant if I have both, if I have red, green, and blue? Those are the colors of the photoreceptors in your eye, that your eye is sensitive to red, green, and blue. So for example, this monitor, which is showing you all these colors, is basically just has pixels that are red, pixels that are green, and pixels that are blue, and by mixing them we can get lots of colors. And that's what we thought as well. Maybe we could get every nerve a different color if we mix different amounts of 3 fluorescent proteins, a red one, a green one, and a blue one, in every single nerve cell. And that would be sort of like we have a green gene, we have a red gene, and we have a blue gene, and then we engineer a mouse, and we call these mice rainbow mice, where we mix together, yeah, not that good, but scientists are not that funny. So for us, it was considered an inspired name, but I know it's not that good. But rainbow was the name we gave this 3-color mixing strategy to turn what would otherwise be on or off fluorescence, just yes or no, yellow or zero, into something much more interesting. So instead of having a black and white image, we would have a color image by mixing together different amounts of those 3 proteins in different cells. And I'm not going to bore you with the technical details of how we did this kind of stochastic recombination, as it's called, to make each cell a different color, but the result was spectacular. So here's a piece of brain. That's the hippocampus at the bottom, and at the very top is the cerebral cortex. And those little dots you see are nerve cells, each a different color. And they're just mixtures of how much red, green, and blue fluorescent protein are in each cell. And if I zoom in on this part of the hippocampus, which is where memories are made, this is the dentate gyrus, you see these colors are really impressively beautiful. It's just a very nice looking color. And if you zoom up farther, you can see also the processes of these cells. These cells in the brain are nerve cells, which are those circular things, and then they have dendrites sticking out in all different directions, and they each have an axon going somewhere. The hippocampus doesn't connect to muscle, but it is part of the central nervous system. In some parts of the brain, this coloring gave us the ability to track axons over long distances. So for example, this is the auditory part of the brain, and these are nerve cells that are sending axons long distances. Some of them are running parallel to the surface, and so you see this mixture of colors, and then some of them are running into the plane. And I'm not sure, can you play that movie? I don't have a control here to play. Yeah, let's see if that plays. He's trying. Yeah. No, I think all of my movies, unfortunately, are not going to play. Anyway, yeah, I'm sorry, this is a technical problem I can't solve right now. This is a movie playing through these cells, but what it gave us was hope that rainbow would be a tool that we could use to trace axons over long distances. And I want to go back now to muscle. If you look at a rainbow animal where the motor neurons, the neurons that make muscle fibers contract, are labeled, they're in the bottom of the spinal cord, and that's what you're seeing in this butterfly image, is just a cross-section of the spinal cord. The cells in the bottom are all fluorescent, and if you follow those neurons' axons out into the periphery, you can, out here, you see this very long nerve now where each axon is a different color. And when I saw this, I got very excited because I thought we had invented a way to let axons or wires be followed over very long distances, until one of my computer science friends reminded me that if you open up any computer, that's what you find inside them, these rainbow cables that were invented a long time before we did this. So this is some kind of convergent evolution, where we now have mice that have wiring that looks like computer wiring. It's very weird and hard to wrap your head around that. If we zoom up on this and look at this at high resolution, the colors are just spectacular. And these colors do give us the ability to trace axons over very long distances. I have a movie of this. Again, you can try to play it, but yeah, I'm afraid we had some technical issues earlier. So these are the neuromuscular junctions. It won't play as a movie, but I think you can see that the neuromuscular junctions are each different colors, and those colors allow us to map the entire wiring diagram of a muscle. And I hope this will show that. This is one axon and all of the neuromuscular junctions it makes in a muscle. All those little ends are the pretzel-shaped objects where it contacts muscle fibers. This muscle has many axons. Here's another axon labeled a different color. And here's another axon labeled a different color. And another one labeled a different color. That one's a little dim. Here's another one. Ju Lu, who did this work, told me that I have to bring the axons in one at a time very slowly because this took so much work that if I just showed it all at once, I would not give him enough credit for the extraordinary amount of effort it was to trace out all these wires. So I'm doing it. I have a contract with him for the rest of his life, so I have to do it this slow way. But it doesn't go on forever. There are only 15 axons, and we're more than halfway. So don't worry, we're almost done. But it is interesting. These axons each connect to a bunch of muscle fibers in a very haphazard pattern. I mean, it doesn't immediately make a lot of sense why it's connected the way it is. Some of the axons have lots of branches, and as we're going through this, you're seeing axons with fewer and fewer branches. And here's the last one, which only has 4 muscle fibers it contacts. So that's the whole connectional map. We call it a connectome of the mouse interscutularis muscle. This is a extremely unimportant muscle in humans. We don't have it, but in mice it pulls the ears backwards, and because we could get the complete wiring diagram, we could ask how similar is this wiring diagram from one example to another, from one instantiation to another. So we looked at the same wiring diagram, did this very same experiment I've just shown you on both sides of the same animal, and we did that for 6 animals. And the result was quite surprising, I think, because animals, like humans, are highly symmetric. There is the mouse. It looks a lot like Mickey in this case. And there are the 2 ear muscles. But the branching pattern of this particular set of axons on the left and right side were not at all alike. And it turned out that every single example, every left was different from every other left. Every right wiring diagram was different from every, God bless you, from every other right wiring diagram. Every single branching pattern was different. Even though animals are highly symmetric, the brain has, in mammals at least, made wiring diagrams of muscle that are highly variable from one instantiation to the next. And that was puzzling, and the question is, why would it be this way? And remarkably, it wasn't just that they were different, they were stupidly arranged. I mean, it would not be the way any engineer who was worth their salt would arrange it. Let me give you an example of some of the crazy things that we saw in these wiring diagrams. On the left is one of these wiring diagrams, and only one of the axons is labeled a different color, and it's labeled red. And zoomed up on the right side of this image is a blow-up of a small region of that wiring diagram. And if you look at the bottom where the axon is coming in, the first thing it does is it makes a straight-up turn where the yellow arrow is, where all the other axons in the muscle go straight. This axon decides to go in a different direction. Who am I to argue that that's not a good idea? Except if you follow that axon, that red axon, all the way up to the top of the image on the right, you see it makes a hairpin turn and comes all the way down again and then goes on with the rest of the axons. It could have saved a lot of distance by just going straight, but no, it wanted to take the circuitous route. Even worse, if you go up about halfway on the left of that axon, the yellow arrows on the left, you see a neuromuscular junction with a little asterisk over it, that little red neuromuscular junction. Again, I don't have a laser pointer, but I think you can probably see which one I'm talking about. And this axon innervates that red neuromuscular junction, but not the way you think. It actually crosses right over, goes all the way up to the top, comes down, and then where the white arrows are, it crosses over itself to innervate that neuromuscular junction, to make that contact. This kind of circuitous route is a waste of axoplasm, if you will. It's a waste of energy to make such long pathways. And it wasn't just that one. If you look at the very bottom on the left, there is another neuromuscular junction of this axon at the very bottom of the image on the left. And that neuromuscular junction comes from a branch that comes off of that red axon. You could have gotten there a lot easier by just going straight where the black arrow is rather than this weird side way of going. And this was typical. There were lots of these inefficiencies, these redundant branches, premature branching, where one branch would do, it would loop around itself multiple times. And this raises a question, you know, animals as perfect as human beings and other mammals shouldn't have problems like this. Where is this variability coming from? And the answer, surprisingly, comes from development. From the fact that the wiring diagram, when you are a baby, or when a mouse is a baby, it's the same thing, does not look like that adult wiring diagram. The adult wiring diagram is the final product of a developmental process that is shaping your nervous system. And this is going to get back ultimately to that first video we saw. So this is two muscle fibers. These are the acetylcholine receptors on the muscle fibers that respond to acetylcholine. And these are the axons that innervate those two muscle fibers. There's one axon on each neuromuscular junction in an adult. Let's look at the same muscle in an animal that is much younger. This is a 1-week-old mouse. The pretzel shape is more condensed. But the thing you'll notice if you're an aficionado of imaging is that there isn't one axon innervating that neuromuscular junction. There's actually two. Two different axons have gone into the same neuromuscular junction. And that means that the nervous system is differently connected when you're young than when you're an adult. And the difference is there's more connections when you're young, not less than when you're an adult. We could, because the sternomastoid muscle is so accessible, we could actually follow a same neuromuscular junction day after day after day by re-anesthetizing the mouse, taking a picture, sewing it up, coming back the next day, taking a picture again. And here is shown at postnatal day 7, 8, and 9, the same neuromuscular junction on the left. It's got two inputs at postnatal day 7. At postnatal day 8, one of those inputs got very, very thin. And by postnatal day 9, it's partially retracted, and now there's just sort of a little ball of axoplasm that's no longer connected, and that's the process of synapse elimination. It goes through a step of atrophy and then retraction. So this is pruning of synapses in a living animal that's normal. This is what normally happens because eventually every neuromuscular junction is singly innervated. To see more of the details, we could make muscles like the rainbow one, or animals like the rainbow one, where each axon is a different color. So here's a yellow axon and a blue axon, and then we followed them over several days. So here is a neuromuscular junction with a yellow and blue axon, and we could ask what happens to the territories occupied by the losing axon. So that's postnatal day 11. We came back and took a picture of the same animal a day later. And you can already see that the yellow axon seems to have gained some ground and the blue axon has lost some ground. Come back a day later. The yellow axon has gained some more ground. The blue axon has lost some more ground. And then we come back a day later. And now the yellow axon owns the junction and the blue axon is retracting away. Come back a day later and the yellow axon has now bulked up, and the blue axon is largely gone from this neuromuscular junction. So axons are competing. They're involved in some kind of competition for land, in this case acetylcholine receptor sites on muscle fibers, and that is happening in every one of your muscles when you're young throughout your body. There is some kind of competition going on between these axons deciding who stays where, and the fact that every muscle ends up with a different wiring diagram suggests that that competition is not preordained how it's going to come out. Something is being worked out as you are using your muscles in early life. Yeah, there's the final picture, all bulked up. If you look at this from the perspective not of one neuromuscular junction, but the branches of an axon, if an axon is making, if two axons are contacting the same muscle fiber, that means each axon must be contacting more muscle fibers in babies than in an adult. And this is a tracing of one of those axons in an adult that's making, I don't know, 15, 20 neuromuscular junction contacts, those little bulbs at the end, in an adult or a 2-week-old animal. In a mouse, you know, a human, a 2-week-old human is definitely not an adult. In a mouse, the wiring is pretty much mature at 2 weeks. The animals already can have babies when they're 40 days old, so they are much older at 2 weeks than you or I are. If we look at the same muscle at birth and look at a single axon, it's just a completely different story. This axon is pretty much trying to touch every single muscle fiber. And when you are a baby, every axon is doing this. That means each and every axon is trying to contact every single muscle fiber. And that means each neuromuscular junction at birth is not contacted by 2 inputs, but by 10 or 15 different inputs. And the most number of connections is right at birth, and that graph shows you that as you get older and older, all this pruning goes on. And when it comes to the brain's wiring, it's all downhill after birth. Your brain is most complicated when you're born, when you know nothing. And then as you start learning things, you become simpler, much simpler. I mean, I remember as a kid thinking my parents were kind of narrow-minded and opinionated. My children don't ask me questions anymore. My children are grown up. They say there's no point asking me a question because they know exactly what the answer will be because they've heard me say it all before. And I say this is ridiculous. I have a lot of information to share with you. I'm an adult. I have all this wisdom. They say you are narrow, you're a dinosaur, you're stuck in the past. And I really disagree with them, except I felt exactly the same about my own parents. And I think this maybe what we're seeing here is some insight into why. It is when you're young, you've got lots of alternative wiring still present, and you haven't yet trimmed all but the right connections, so a lot of your ideas are wrong. And then when you get as old as me, you're fortunate that you only have wisdom left. You only have the right connections left, although a child may not be able to appreciate that so much. Yeah. So here is the what's going on in muscle. At birth, you start out with an all-to-all connectivity. Every neuron in the spinal cord at the top is trying to touch every single muscle fiber. And then you have this competitive set of mechanisms going on where each axon loses some connections and each axon wins and becomes the soul mate. Literally, for the rest of the life of the animal, that axon and that muscle will remain together. From the muscle perspective, it's a monogamous relationship in the sense that there's only one axon left. The axons are still playing the field in the sense that they have multiple partners that they remain in contact with. I am not making any implication about which is the man and which is the woman. It wouldn't make any sense to talk about muscle that way anyway. But it is interesting that there is a kind of courtship going on where the axons are competing for the right to remain connected to a muscle fiber, and one wins in every single case. So what are the implications of this kind of profound rewiring? First, mature circuits of nerve cells, and I've just shown you muscle, but this is true everywhere we've looked, emerge after a period of pruning from the much more interconnected nervous system of juvenile animals, including human beings. Pruning is mediated by neural activity. Sensory experience and motor practice is what's deciding who stays and who goes. And you become, we become based on what remains after the pruning. You would not trust a 2-year-old baby in a house alone because they are not yet fully formed. A dog by the age of 2 years is much more trustworthy than a human at the age of 2 years. One of the special things about humans is we go through this process much slower than any other animal, giving experience a much greater time in which to shape our nervous systems. People, even as old as me, claim that I'm still capable of learning, although in my household there is some debate about whether that is true or not. So we start out perhaps with the potential to be many things, and each of us sadly ends up as a small subset of those, maybe because of the decisions we've made as we grow up, the teachers we've had. And remember, what this means is education, which is sort of changing the wiring diagram, probably by simplifying it, is removing connections. In this sense, education is destructive. You start out with all this potential, and then education tells you to get rid of a lot of it. I, as a teacher, don't like thinking about myself as a professor destroying the brains of my students. I say I'm pruning properly, but I better be telling them what's actually correct, and I believe what I'm telling them is correct, but truth be told, I have no idea. So these are problematic things going forward. I want to end with one final movie. And this is an interesting movie, also from the internet. It's a 4-hour time lapse of a 9-month-old baby who can't yet walk. And I think it will tell you why this notion of choosing wiring is so fundamental to being a human being, because we explore the world with a kind of crazy compulsion that no other animal has. So please watch this. This is time lapse, by the way. It won't take 4 hours. That's a mirror it's looking at, bouncing up and down. There is no object in the room that this little kid doesn't touch. Like this cardboard coming up here, this box. See that? At that point, he got trapped underneath a chair, and his parents nicely got him out of that mess. It's kind of amazing to see that, because we don't normally think kids are doing much when they're touching objects and twirling them, but they're basically memorizing the shape of every object they've seen from every orientation. People who have no sight as children because of opacities in their lenses, when they are given sight as adults, are mystified by the visual world. They don't make much sense out of it. And one person who was given sight as an adult when the lenses were corrected said, I do not understand how people who are sighted understand that that's a chair, because from every single direction it looks different. How do you know those are all chairs? And we say, well, it's trivial, but it's not trivial. It requires an extraordinary amount of learning about the world. And it probably comes about through a pruning of a wiring diagram that is designed to make you compatible with the world you find yourself in. And that's kind of a remarkable thing, because the world is changing very rapidly. People like me are called dinosaurs by my children. And my children will be called dinosaurs by their children if they have children, and so on and so on. This is just a remarkable thing about human beings. We will adapt based on this remarkable ability to change our wiring diagram to match the world we find ourselves in. So that's all I have to say. Happy to talk, and sorry some of the movies didn't play.
Let's give him a round of applause. Thank you very much. All right, everybody, this is audience Q&A time. The way this will work is that I've got a microphone, Katie has a microphone. Raise your hands, we'll bring a microphone to you. That way everybody in this room and our overflow room can hear the questions and be able to participate in the discussion all together. So I think Katie's headed to the first one.
Sorry.
And keep waving us down. We'll be looking around. I see some hands going up in the back, so make sure you keep flagging us down so we can get to you.
So we saw the 6 mice with their 2 sides, and the wiring was all different. And I think that your explanation seemed to be later on in the talk that they all had different experiences, and so the pruning happened differently, which seems plausible. But I'm wondering if it's also possible that the environment facing these mice was not so challenging that a kind of evolutionary fitness was required, so that, you know, the one most fit wiring pattern would have emerged to allow survival and reproduction. How do you distinguish those two explanations?
Now, this is a very good question and a very subtle one. If you look at animals that do not undergo this developmental rearrangement, which are animals like insects or worms or fish even, you find wiring diagrams that are absolutely stereotyped from one animal to the next. And one way of thinking about that is that it's very easy for the nervous system to encode a particular set of connections a nerve cell should make. It's somewhat problematic not to do that, actually, because there's a cost in not doing it. In the case of humans and young vertebrate animals, they're kind of helpless, and humans remain helpless until they go to college, at least. I'm hoping that's, you know, that it takes a long time. But there's cost to not dictate the right answer. The advantage is that the right answer may depend on the environment you find yourself in, so you want to give the nervous system an opportunity to change. But most animals on the planet do not do this. They are absolutely exactly the same. So for example, if you wanted to get a whole wiring diagram of a fruit fly, you don't have to get every single axon in every single animal. You can get one axon from one animal and take another axon from another animal and superimpose them, because they're in stereotyped so much that they're exactly the same from animal to animal. So that is why we think it occurs this way. It's not that there's a cost, a fitness cost that's profound. You know, babies can't turn over. It takes human babies a year to walk. This is crazy. There's no other animal that takes a year to walk, and that's all because the wiring isn't figured it out yet, whereas most animals know how to walk. But, you know, it would be hard to make most animals do the tango or do pole vaulting. And, you know, we can do all these extra things by virtue of having this plasticity.
So, first of all, thank you. That was, as a physics geek, I had no idea biologists were having so much fun. So that was that was great. So, as you mentioned there, humans, we have this insanely extended childhood. Does anybody have an explanation for what's going on in the brains of adolescents?
No.
Next question. Yeah. No. I mean, no one really knows what's going on in the brains of adolescents, but it's not so clear. It's not actually clear when we grow up, if at all. Humans are much more immature. It's called neoteny. We maintain juvenile traits well into adulthood. And adolescence, you're clearly not a mature animal. I mean, we don't allow humans. It takes a year to walk and another 15 years before we give people a driver's license, at least in Massachusetts. I don't know, down here maybe it's a little younger. No, 16. And then you leave the nest 4 years later, and you're still not an adult. You're in college. And if you remember college, if you went, you're a child, definitely. And then it's another 10 years or so before you get your assistant professorship. And then another 15 years after that before you get tenure. And another 10 years, now you have gray hair, before you're a professor. I mean, it's insane. It shouldn't be that you have to be over 50 before you finally have tenure. But that's the way it works, and it is this very slow ramp. But adolescents are still very young children. They don't think they are, but all evidence suggests they are. Yeah.
I think it's at Harvard where they have that connectome study being done.
At Harvard?
I believe it
Yeah.
is. And if
I'm part of that.
Oh.
Yeah.
If you look at, as you described, the variability of this pruning process, doesn't that make that connectome project really difficult to achieve, to find all the connections?
Well, we can get the connections. It's just every single example is completely different from every other one. So why is that a problem?
Because when you study the connections in one brain and try to match them up with another, they're always going to be different.
They're going to be different, but they're probably not going to be different in a random way, because they're all doing the same thing. They're encoding information about the world. So there must be some regularities to the way information about the world is wired into the nervous system. And earlier today I talked about this connectomic work in the brain, and it's extremely depressing to talk about because it's so complicated, and even doing one brain requires a human brain would require a digital content that exceeds the digital content of the world right now. So I don't have enough storage to do that. Every cubic millimeter of brain is 2,000 terabytes of data.
Wow.
Yeah.
Thank you. That was a fascinating presentation. I got two questions. The first is, how do the axons recover from an injury? And the second would be, during pruning, I understand that there's external catalysts or stimuli that's driving the pruning, but what is the actual characteristic of the axon that makes it preferential over another one? Thank you.
Yeah, both very good questions. So in the peripheral nervous system, that's axons that leave the spinal cord and go out into the body, or axons that leave the nerves in the brainstem and go out to the face. If those axons are damaged, they grow right back to where they were before, because there are tubes made of glial cells that they run down. And I didn't bring movies of this, although none of my movies played anyway, but had I brought them, you would see that an axon would go right back to that pretzel-shaped acetylcholine receptor area and reoccupy all those sites. And they go fast. And it turns out that often this is happening to people all the time. My brother, when I was a kid, used to give me something called noogies. I don't know if you know this, where they into my arm muscles, and probably denervated muscle fibers all the time, but the axons would just grow right back. If you cut a nerve so it can't find the distal tubes, then everything grows very poorly. They grow back, but they miss a lot of areas. Unfortunately, in the central nervous system, if you damage an axon, it doesn't know what to do. And they grow just very small distances and get lost almost immediately. And that's why spinal cord injury is so devastating. The other question was about synapse, what's happening to these axons, I guess. Yes, who makes the decision? And for a long time we were unsure, and now we believe that the decision is being made by the muscle. The muscle is listening to these axons based on the amount of neurotransmitter they release, how often and how much, and then it reinforces one by adding more receptors under that one and begins to pull receptors and adhesive molecules away from the other one. So the other one still wants to talk to the muscle. It doesn't want to give up, but the muscle's got like cotton in its ears. It doesn't hear it so well, and that starts a vicious cycle where one gets stronger and the other gets weaker, and when the other one, the weaker one, no longer can even find a place to hold on, it just pops back and resorbs that branch. It's not a tragedy, though, because every axon is losing branches. They all start out with almost everything. So all of them are losing some connections and strengthening others. They're all just sort of reapportioning their synaptic drive to strongly do a few things rather than weakly do many things. And it's exactly a sort of analogy to what happens to human beings during development. We start out dabbling, dilettantes, trying all these things not too seriously as kids, and then we decide, I want to be an electric guitar player, and the parent says, oh, don't do that. But that's what they want to do, and then they put all their energy into that, and they give up stamp collecting or whatever else they were doing. And I think that's exactly what's happening inside your body, and the reason these things are analogous is because you are just the product of all these processes going on inside your body. It's the same brain doing both of these things.
Thank you very much for the fascinating talk based on your research. And when you were talking about how mice have muscles that can pull their ears back, a question just flew into my mind. It's a very difficult question. When I was a kid, I noticed that there are some kids and adults that can wiggle their ears.
Yeah.
And my father could. I couldn't.
Yeah.
And a lot of adults and kids can't. My question is this genetic or is this pruning?
You mean, did we all start out with the, it's a good question. Could we all prune, could we all wiggle our ears as babies? And then most of us smartly got rid of this stupid, obnoxious trick, and a few people held on to it because they got positive reinforcement from their older brother or something. Because my younger brother can still wiggle his ears, and I remember him saying, wiggle your ears, and I would be laughing at him. But I can't wiggle my ears because no one ever asked me to do that. So maybe you have it right. It never occurred to me before. That's a brilliant explanation for the variability here. Not genetic, it's environmental.
Oh, okay, she is a little bit chicken, but she is wondering, her dad grew up in India and her mom grew up in the United States, and so have you noticed in human brains, are there any like cultural, geographical differences? Like, are Asian brains more wired a certain way as opposed to American brains and European brains?
I don't know, I have no data on this. But given the fact that, just as in this audience, most of the places I visit and where I teach, you have a diverse number of people from a diverse parts of the world, I think humans are much more similar on the insides than the outsides. And an example of this can be seen in lions and tigers, which look completely different from each other. But if you took the skin off, or if you painted them white and shaved them, you'd have a hard time telling the difference between a lion and a tiger. That's a lot of our traits are related to evolution of our relation to the world around us. And as you go farther and farther into the brain, we become more and more similar because there's less sort of environmental input to the way the center parts of your brain are. We see this in worms, worms that look very different on the outside. When you look at their wiring diagram, as you move in towards the center of the worm, they're all identical. So I would suspect all human beings are pretty much the same as you move in. That would be my guess.
Let's give him one more round of applause. What a fabulous talk. Raise your hand if you learned something. Okay, good. That means that you should probably come to next week's Science Cafe as well, right? You can expect to learn something there too. Science Cafes happen here at the museum every Thursday night. We bring in scientists, special guests to the stage. You can learn. Next week, next Thursday night at 7, we won't be in the Daily Planet Cafe though. We will be in the WRAL 3D Theater, so the first floor of the other building of the museum, just up the road here, for Dr. Lee Gatkin, How to Tame a Fox and Build a Dog. If you're familiar with the Siberian research on taming foxes to learn about the evolution of canids, you don't want to miss this special lecture with Dr. Lee Gatkin and book signing. That's next Thursday at 7 o'clock. You can head on into the first floor of the museum, check out some of the exhibitors that are still here. Thank you all so much. Have a great evening.